Good morning. I hope everyone can see the screen there.
Looks good. Yep.
Perfect. Thank you. Great segue into the call there, Taylor. The Zoom meeting and an action-packed agenda, and we're going to move through the forest health conditions for Ontario. But before I do that, I want folks to stop for a moment and think where we were a year ago today. It was just coming on to Valentine's Day. We were all gathered in the beautiful Allison location, the Nottawasaga Inn, and we were talking about forest health and other occurrences on the landscape and forest management. And then all of a sudden, we were talking about the weather. We were talking about what's going on, and we're following the news a little bit. And very shortly after that, the world basically stopped turning. And when it stopped turning, the first thing that people noticed were the trees. Last year the theme of the Forest Ontario AGM was We the Forest, and we looked at that and moving into this year, growing our forest, and there's a lot of really bad times that have come over the last year, but there's been a lot of really good things that have occurred as well. And I think the moving, as Taylor's mentioned earlier, the science and the importance of science and the importance of trees in our green spaces on the landscape, and what they've provided to us as the people of Ontario. So as we go through, I want you to sort of think about what I'm talking about from a pest and disease side of it. We had a lot of eyes on the landscape out there, and we had a lot of people paying attention to what's going on this season. This is going to be a snapshot of what went on in the province and a little bit about our forest health program. So as we move through growing our future with the theme from the Forest Ontario Conference, this is how we're going to grow our future. Our forest is so important to us all now. We all have basically been snapped back to reality. So my name is Dan Rowlinson. I'm with the Ministry of Natural Resources and Forestry. I'm part of the Biodiversity and Monitoring section. We're a very diverse, how do you like that pun, a diverse group across the province that we have different parts of our program that monitors the fish, the wildlife, and my program from forest health with the trees. We have a number of collaborations and partnerships that we depend on to deliver our information with the Invasive Species Center and the Canadian Forest Service to look after our diagnostic service that we have. So when we say we're collecting something like gypsy moth on the landscape, we have it verified, and that information is credible. Through the Forest Research Institute here in Sault Ste. Marie, we have support through Sylvia Griefenhagen to do our disease work, our disease diagnostics. And we have the management team within BAMS that supports the crazy ideas that Dan comes up with every year to monitor and to provide this information that we're collecting. And I mentioned Dan Licks here. He's our GIS guy. And with everyone who's been involved in creating presentations and reports knows that if you don't have a good GIS support, it's very difficult to get that data together. So a great acknowledgement to all my staff that are involved in this, and of course my program staff that were forced to deal with a number of thinking on their feet exercises this season when it came to monitoring. The way we're structured, we have a crew in Northwest region, Northeast region, and Southern region. So we're just disseminated across the province with each individual being responsible for a work area. Going to start my talk off with, it's almost like a sunrise when you start talking about forest health. Forest tent caterpillar always holds a special place in a lot of folks' hearts because they're a disgusting insect. 2019, we had 35,000 hectares, and we look at the graph of the data over time, and we know you can almost set your watch by the typical outbreak cycles. And in 2020, I'm happy to report that we did not map forest tent caterpillar damage or detect it anywhere in the province of Ontario. We did have a couple populations that were popping up in the early spring, but moving into the field season, the natural diseases and parasites have caused that population to collapse. So forest tent caterpillar, thankfully, as we will learn going forward, and we're dealing with the gypsy moth, forest tent caterpillar is no longer an issue currently in the province of Ontario. Spruce budworm, mainly a pest of the boreal forest. We have noticed populations in southern region as well. There are, historically there has been defoliation. It's this season moving into 2021. It's the root of a planning exercise to have a potential operational control program to protect the foliage in Northeast region on some high value stands. Our data on this graph dates back to the 50s. We see those typical cyclic outbreaks. And as I show these graphs, I want you to sort of consider also the technology that's moved along as we've collected our data in the field through the Forest Insect and Disease Survey program that was housed within the federal government. The technology of the day was the best it could be. It was the aerial mapping or sketch mapping. So that data sort of, when it was collected in the field, was more of an overall perspective of the defoliation. So we're seeing, in the late 70s, early 80s, defoliations ranging into the millions of hectares. And we move into our now way of collecting data in the field with a more refined method of digital mapping tablets. So we're able to get a more refined picture of what's going on in the landscape. 2019 spruce budworm was roughly about 350,000 hectares of defoliation across the Northeast region and just down starting into the northern part of the southern region. 2020, we've seen that expand considerably with about 442,000 hectares of defoliation across the landscape and mainly in the northern part of the province, but as well moving into the top end of that Parry Sound district in southern region. So this has triggered the management initiative on the Crown Forest to develop a plan to deal with it and provide some foliage protection in these high value stands. Jack pine budworm, it's another cyclic insect that we see. These are native insects, by the way, and I want to make sure that that's highlighted as well. But another budworm on the landscape, we've been dealing with this one in the northwest part of the province for the last number of seasons. It's been the root of 2019. There was an aerial control program of just over 100,000 hectares of jack pine forest that was sprayed with Bacillus thuringiensis BTK to provide that foliage protection and protect some of those high value stands and the resources and also to provide some protection from fire in those far north communities by reducing the mortality that's out there. Again, a relatively cyclic insect in the province that we've seen in the 80s, where the province sprayed hundreds of thousands of hectares of forest with that range of defoliations seeping into the 4 million hectare range. And in 2020, this season, 2019, we had just over 1 million hectares of defoliation in the northwest part of the province. Moving into 2020, we're starting to see a bit of a reduction. The defoliation is breaking up somewhat. We have just under 1 million hectares, about 930,000 hectares of defoliation in the northwest part of the province, moderate to severe. And we are planning a control program in 2021 of about 100,000 hectares of jack pine forest to protect those high value stands and keep those trees green while we start to see this outbreak collapse naturally. One thing that when I talk about forecasting or the work that we do from a forecasting perspective, we've providing support to the management initiatives. Jack pine budworm, we've carried out a second instar larva survey. And this sort of gives us an idea of what the population is moving forward into 2021 as we plan a management response. We've done the same for spruce budworm, and we've also carried out a forecasting initiative for gypsy moth in the southern part of the province. But what I want you to look at this map is you see all those green dots. If you recall back when I was talking about the defoliation, you can see that sea of red. And you're looking at this and you see these light forecasted populations. So as we move forward, we are starting to notice that the decline in the natural decline in the population of jack pine budworm is starting to show itself on the landscape. So what is a forecast? Whether it's spruce budworm or jack pine budworm, the overwintering survey is when we go out in the forest in the wintertime, we collect upper mid-crown branch samples, we bring them back to the lab, we basically burn the little suckers out of their overwintering location, and we count them under a microscope. And using that information, plugging that into a formula, gives us an indication of what to anticipate for defoliation in 2021 or in the next year following, and that will give us an idea of where we should be directing our control initiatives. This gives you an idea of what the lab process looks like. We soak them in a solution of sodium hydroxide, and that's what burns the hibernacular, the tiny web, off the larva. The next, lots of folks across Southern Ontario might have had some experience with this pest this season, the gypsy moth. It's an invasive species on our landscape. It's a defoliator of oak, birch, and all things hardwoods, with the exception of a red maple. Doesn't like to eat red maple. And you'll also find it consumes the foliage on the white pine trees, and it was a real pest this season. And I'm sure I will get no argument out of anybody on the line who's had a coffee on their back deck in the morning within a hardwood stand that was infested by gypsy moth. Interesting, 2019, we had a persisting defoliation level moving across the southwestern part of the province. We had about 45,000 hectares in 2019. It's been bubbling away since the 80s, and we refer to gypsy moth as, although it's an invasive pest, it is seemingly naturalized in across the landscape of Ontario since its outbreak status in 1980. As the evolution of the larvae have moved along, we've seen these parasites, predators, diseases that have been able to evolve within that population and cause this sort of natural cycle across the landscape. You'll look at that 2020 defoliation, where it moved to almost 600,000 hectares, sorry, in 2020. It took a real big spike. 2019, down in the southwest part of the province, that's where we've seen the bulk of the defoliation. There were numerous control programs that were conducted across the southwest by municipalities and private landowners to carry that, to try and keep those trees green through the outbreak. In 2020, we've seen that gypsy moth infestation expand considerably in the province to just over 560,000 hectares of defoliation, extending all the way up to the shores of Lake Huron in Sault Ste. Marie district, and we have quite a significant area around Sudbury that has caused severe defoliation to the oak ridges there. Looking at this map, it looks like there's a lot of red out there. But if you look at that down in the southwest part of the province, where that infestation is starting to show its age, and that information has manifested itself in the egg mass survey that we carried out this season. We are seeing still a severe forecast, but we've noticed high parasitism in the egg masses down there. So we would anticipate that insect is going to start showing signs of decline moving into 2021 in that southwest part of the province, where the infestation is entering its 4th and 5th season of severe defoliation. Gypsy moth is a quarantine insect. It's a quarantine under the Plant Protection Act by the Canadian Food Inspection Agency. A lot of people have seen it so far widespread, they don't really realize that it is still a quarantine insect, and it's managed by the Canadian Food Inspection Agency in Canada. Forest Health Monitoring Program does support the initiative from to support the quarantine of the gypsy moth. We support that by trapping with the CFIA, collaborating with our partners to monitor gypsy moth populations outside of that area of quarantine, placing these pheromone traps that will capture male moths in the shoulder season. So when you're mapping these disturbances, I like to give people sort of a perspective of what it is we're actually putting that little red dot on the map. We fly over these stands and grid lines. We look down out of the aircraft. We have an observer on both sides of the aircraft with a digital mapping tablet. And we look at these stands, and this one is obviously at the severe defoliation level. This gives you a perspective of what went on over in the Grand Bend area this season. This is a view of one of our flight lines from the aircraft. So fairly significant defoliation that we're observing. This gives you an idea of the startup of gypsy moth, where it likes to get its foothold, defoliation on the hilltops. That's where you're going to start to see that landscape level defoliation start, is on those higher elevations where the moths move in and they start their defoliation. So we talked about having the support of the Invasive Species Center. So we go on the ground after we complete our aerial surveys, and we collect specimens of the causal agent. We send it in, so that is recorded in the database of what it actually was. Gypsy moth egg masses. So we talked about forecasting for the other two pests out there, the big ones this season, the jack pine and spruce budworm. The tool that we use for the gypsy moth, is the egg mass survey, modified Calidar plot, or commonly referred to as an MKP. We also look at the virus loading within a stand. These inverted V's larvae hanging on the trees. gives us an indication that there's a virus in that population that could be having an impact on the outbreak. And if there's one thing we've learned in 2020 is the impact a virus can have on a population. And whereas and I'm sure everybody's heard these analogies, but in our population, we want folks to be wearing a mask, social distancing. When it comes to the gypsy moth, we want them climbing all over each other, spending Christmas with each other, and spreading that virus around, because that's what's going to cause that natural collapse of the population. So we did initiate a survey this season with the modified Calidar plots based on 10 square meter blocks in the stand. We go through these stands, we count the number of egg masses on the ground, we count the number of egg masses above the ground, so on the trees, the main stems. We plug that into a formula, and it gives us a prediction of the 2021 defoliation. And that information is all available through Ontario.ca. So if you Google Ontario.ca, it was just the forecast map was just loaded this week. So we have that information there, and we're working on a frequently asked questions document to be posted there as well. But you'll get an idea of the forecast of gypsy moth throughout the southern region, and it does give you a severe forecast, but you'll see where those plots were put in. We also look at the size of the egg mass, whether they're loony size or their dime size. So that gives you an indication of the age and the health of the gypsy moth population. One of the other hardwood defoliators that we had across the Northeast was the large aspen tortrix. And it seems to go hand in hand with the forest tent caterpillar. And now that the forest tent caterpillar outbreak is collapsing, we did have a large number of calls this season regarding a moth flight, and these early in the season, and these moths were definitely tied to the large aspen tortrix outbreak that's going on in the northeast part of the province. And again, this gives you an idea of what we were looking at from a northern perspective when we're looking at defoliation across the landscape. So fairly widespread in the hardwood stands. And this moth compares on the hardwood leaf. You look at the moth of the large aspen tortrix, and on that balsam inset image, you can see what the spruce budworm moth looks like on there. So they're fairly similar. Defoliation, unlike forest tent caterpillar, large aspen tortrix gives you a sort of a ragged appearance on the tree, not a very messy feeder as compared to forest tent caterpillar, or gypsy moth, that totally consumes the leaf. White spotted sawyer beetle, I bring this to your attention. We had a couple small areas in the northeast part of the province of white spotted sawyer beetle damage. It's one of the insects that was commonly confused with the Asian longhorn beetle. It's an insect of pines, attacks stressed trees, whether by fire, drought. It's a pest of conifers. And another good announcement that we were just coming up to was last season was the announcement of Asian longhorn beetle being eradicated from Ontario, and white spotted sawyer beetle is one of our native pests that we still see across the landscape. Gives you an idea of some of the damage that it causes on these stressed trees. You can see all the sawdust on the ground. This is in a fringe of a recently burned area in Northeast region. Oak decline. We did have some reports of oak decline in the province this season across the north shore of Lake Huron. Again, another one that we investigate fully because of the threat of oak wilt on the landscape. We want to make sure that these calls that we do receive or that do come in from the public are not oak wilt. And we rely on that citizen science perspective to support some of the survey work and direct our surveys that we do on the area. This gives you an idea of the what was found in that stand. It was armillaria root rot in a relatively shallow soil site, which was causing the mortality and the decline in these oak stands. So not oak wilt, but again, something that we certainly want to investigate. One thing that I mentioned when I started off was the virtual world of forest health, and it really became evident this season that there were a lot of eyes out there on the landscape, and there's a lot of really good photographers out there. And when you're sending images in for identification, or you're asking questions of do you know what this insect is, or should I be worried about it? When you're sending pictures in, an ideal image of the larva is excellent to get, but also a picture of the damage. The damage is important as well. If you look down in the bottom left corner of the slide, you'll see definite white pine weevil damage to a jack pine area. On the right, you'll see some defoliation to jack pine. That's jack pine budworm. Up in the top right, that telltale line of sugar maple borer. And again, we have lovely larval images that come in. So again, the virtual world of forest health was alive and well this season, and it really helped us to sort of direct some of the survey work we do and provide our field staff with some investigation areas to investigate throughout the field season. And we were started off our field season very slow, and we weren't sure whether we were going to even have a field season, but we were able to get protocols and safe working procedures in place in time to carry out our aerial survey work, and that's why we were able to produce these maps that we have this season. Again, planning into 2021, we're certainly anticipating to have staff in the field a little bit earlier this year. One thing we miss when we're not out to the field later is these sort of verification surveys and getting an understanding of what's going on in the landscape. And so we depended a lot this year on reports from the public of these brown trees or defoliated trees to sort of guide some of the survey work I do. On that note, I'd also like to mention Ontario.ca, where you'll find our forest health conditions report. That's the sort of the full compiled report of the forest health conditions in Ontario. 2019 is loaded. We're currently in the process of writing the 2020 report, and with anticipation of it having it up and running in mid to the end of March. So Ontario.ca, and also visit that site for all your information on gypsy moth, the maps, the forecast information that's out there. And on that note, I thank everyone for their for joining in today. And again, remember that how important growing our forest is to all the people of Ontario. Thank you very much.
Thank you, Dan. I think since you finished a little bit early, we're going to take a couple questions now quickly, because there were a few that came in through the chat, if you don't mind. So the first one I see here is a two-part question. Who is coordinating the lack of GTHAA data on gypsy moth in Ontario to incorporate the approximate 450,000 hectares of 2020 infestation, so we have a true picture of the extent of the significant infestation on the horizon?
Okay, good question. So again, I'll sort of draw your attention to that Ontario.ca, where you can see the map, you can see the forecast information that is out there. Also in the forest health conditions report, you'll see summaries of the level of virus loading that we're seeing in the population, the level of parasitism. As I mentioned, although the province is not does not have the initiative, or not the initiative. We have initiative. We just don't have the legislated ability to control pests on private land. There are going to be a large number of spray programs that are conducted by the private sector out there this season to protect municipalities, provide that that protection to foliage on parks, and in areas like that, sugar bushes. So there are going to be large control programs that are conducted by the private sector. But I would direct folks to that Ontario.ca website, gypsy moth, and I believe Nicole has shared the link at the top there to get that information moving forward.
Awesome. So the second question was, how is the province coordinating a media outreach to prevent confusion of responsibilities in the various levels of government to more effectively and efficiently deal with this important pest?
Yeah, another great question. And I mentioned, as I mentioned in my sort of onset there, the development of the Q's and A's that we're working on to get a uniform message out there. And then we will be linking that site through with our partners, particularly, the Invasive Species Center that's done a really excellent job of getting the message out there for folks. Through our other partners, the Canadian Food Inspection Agency, the Eastern Ontario Model Forest, that information will all be shared and linked through those sites as well. Also, the question on the egg masses on Colorado spruce. One thing that gypsy moth will do is that it will lay its egg masses on absolutely everything. And I'm sure people that have experienced a gypsy moth outbreak this season have noticed that. Just because there's an egg mass on that Colorado blue spruce doesn't mean it's going to be defoliated right away. Remember that the priority host for gypsy moth, in order to get the nutrients they need to get their foothold in the spring, they need those nice green oak leaves. That's their primary host. So as the leaves start to unfold in the spring, the larvae are hatching, they move in, they start that shot hole appearance feeding on the leaves, and that's how they get established. Once they totally consume all the hardwood foliage, that's when they move on to these other species, like your white pines, your ornamental Colorado spruce, your white spruce growing in your yard. So that's important to understand as well, that they primarily feed, their preferred host is the oaks and the hardwood species, but then they move on. Once they've eat themselves out of house and home, they start to move on to the other species that are intermixed and growing within these other stands. And they'll eat flagging tape. They'll just about consume anything that they can climb up on and consume.
Awesome. So we'll do a couple more before moving on. Is MNRF or CFS preparing to aerially spray gyp check to reintroduce the virus to the landscape as they have done in the past?
So speaking for MNR, MNRF does not plan to do any spray programs for gyp check and the virus. One thing I also will bring to you, and maybe Taylor could add a message, or Chris in his discussion about gyp check on gypsy moth. But remember that virus is always present in the population out there. So those gypsy moth that right now are sleeping in their nice little warm egg masses, there's virus in those gypsy moth larvae. As they come out and they start feeding and they start spreading that virus around, that's how that virus is going to be spread within the population. And the population that we see on the landscape right now is so intense. It is a perfect storm for a virus outbreak to take a foothold and cause that collapse in the population. As the larvae feed in the upper crowns of the trees and they die from the virus, they almost explode, and this virus spores sort of cascade down to the other lower feeding larvae, and then they climb over each other, and there's a grooming period in the day that these larvae have as well, and that's how that virus gets spread. So the virus is very effective at controlling populations, in combined with the other factors like the extreme cold days that we may have of egg masses above the snow line, for example, that infestation that's up in the Sudbury area, and as well the number of parasites that are attacking the egg masses right now, and as I mentioned, in that southwest part of the province. So all these factors combined are going to be the have the responsibility of taking knocking that gypsy moth population back down to a more manageable level. Long answer to a quick question, Victoria, but
that's all right. So was there any forecasting for gypsy moth done in the Parry Sound or Sudbury districts?
So we did not do the Sudbury area. We did do in the south of the Parry Sound area. So you'll see some egg mass locations, and there is a severe forecast in that area. You'll see a few egg mass locations on that map that are within that area as well.
Okay, and then somebody's asking if they can get a copy of the gypsy moth chart. It doesn't appear on the Ontario website, and they feel that it is an important part of GM communications, gypsy moth communications.
The graph?
I think that's what they were asking for.
If it is a graph, so we've just completed these graphs now. If you send me a direct message, I can certainly share that with you.
Yeah, and they're confirming it is the graph.
Yep.
It is the graph they're looking for. We'll do one more before we move on, because we do have a couple more minutes here. On the gypsy moth forecast map, it can be seen that in 2021, there is presumed to be moderate defoliation in my local municipality. At what level of defoliation is a spray program admissible?
So that's a really good question. That would take me probably most of the rest of the morning to answer. But so moving forward through a what we call an integrated approach to pest management, remember spraying is only one tool you have in your toolbox. There's the other components combined as well as, healthy growing, vigorously growing trees can withstand a few years of of severe defoliation, as long as we don't get drought or other diseases that may come in and impact those trees. So if you're using something like a spray program as a tool to manage the populations, you got to keep in mind that that's only a 1 year approach to pest management. You go in there, you spray, you're just providing that tree foliage protection for that given season. It's not like a can of Raid where you go into your, to kill a hornet's nest, you spray it, they're dead. BTK has to be ingested by the larva, and it has a very short shelf life on the foliage. It's not something that's going to remain for months on end. Once the gypsy moth consumes it, it crystallizes in the gut wall and it causes that collapse. So again, getting back to your question of what triggers an approach or a spray program in your municipality. Well, it's going to be what your management objective is for that area. If it's foliage protection to keep a park green, then that's defoliation levels at, that 50 to 75% would trigger some type of control program, whether it's an egg mass removal or it's keeping these trees green through the employment of something like BTK or bucket spraying.
Awesome. And Nicole's going to bring in a question from the Q&A.
Yeah, Dan, just one more before we let you off the hook here. I've got someone in the question and answer section, Debbie, asking if the province has any help or support with funding for spray programs. So be it for gypsy moth or any other programs that you're aware of.
Yeah, I'm not aware of any support or funding initiatives at this time that are run through the Ontario Ministry of Natural Resources. Depending on what what it is you're spraying, whether it's if it's a plantation area, the Ministry of Agriculture Food may have something there for assistance, or it's a nursery, something like that. So it would be worth checking with them as well.
Awesome, thank you. So 2 comments before we move on to the next presentation. Chris MacQuarrie is saying that gypcheck is no longer produced. And we also had a comment from Peter Winizuk, sorry if I messed up your last name there, that there's no flyover in the GTHAA due to Pearson Airport. Therefore, there is a considerable gap in information. But we'll move on and we'll come back if you have any further questions for Dan. We'll come back to those around 9:30. So continue to ask them if you have any follow-ups, and we'll try to get through as many of them as we can.
Good morning, everyone, and thanks, Victoria and Nicole, for the invitation to present this morning, and to everyone for taking the time to be here today. So I am going to just make an adjustment. I'm going to talk about our research on biological invasions. And today I'm going to focus on plant microbe interactions and how they can affect the trajectory of invasive plant populations. So alien or non-native species are those species that are introduced as a result of human activities. And some alien species, they become established and they form self-replicating populations. Now, some of these species, they start spreading very rapidly over large distances, and they cause some form of detrimental impact. And this is what we then call an invasive species. And as you can see in this map, alien species, and this map is put together with vascular plants, ants, spiders, freshwater fishes, amphibians, reptiles, birds, and mammals. So in this map, alien species, you can see they are widespread around the world. And, together with what this tells you too, is that together with things like plastic pollution, climate change, urbanization, this is truly a global change factor. So, and the other thing we can take out of this analysis is that Canada's provinces are in the upper 50 to 10% of regions with most alien species. Now, the problem keeps getting bigger, and this can have far-reaching consequences, both for biodiversity, ecosystem level function, and the emergence and spread of existing and novel diseases. So today, I actually want to focus on biotic interactions between plants and diseases, soil biotic interactions. That's what I'll focus on today here. And so, everyone working with on invasive species is familiar with the invasive species curve, right? So basically, after an invasive species is introduced, there's a lag time before it starts growing exponential. And then at some point during that expansion, the species is recognized as an invader, right? So, but at that point, somewhere around here, it's usually too late for eradication, and so costly control is then all we have where we're left with. But with so many alien species to choose from, now how can we enhance our capacity to predict invasions, and perhaps even more importantly, assess their persistence? Many hypotheses have been proposed to explain why species become invasive, and these have been actually recently very nicely synthesized in a book, and also on this corresponding interactive website, where you can actually click on each one of the hypotheses in invasion science and see how much support they have received through empirical testing in the literature. So if we zoom into the hypothesis around biotic interactions, what I want to point out here is that even new associations, the new associations hypothesis focuses on those early stages of the invasive species curve, so around establishment, rather than focusing more on the later stages of invasion. This is pretty much true for most of the hypotheses around there. So you see here, new relationships between non-native and native species can positively or negatively influence the establishment of non-native species. So, but then what happens to, and today earlier on, we looked at the gypsy moth, and so we had a glimpse about this. Today I'm focusing more on where we are with plants. The thing is, invasive species, they are expected to encounter organisms that over time will learn to utilize them as a resource. So if this is true, and we can find a way to then predict which invasive species are more susceptible to disease, this may help prioritize management practices. So what is the evidence then for pathogen susceptibility in invasive species, and how can we predict pathogen susceptibility? So one of the first invasive plants that we started working with here in our lab was dog strangling vine, or Vincetoxicum rossicum. That's a photo of our group when we went down to Southern Ontario to some of the more around urban forests and parks, and around cities. I mean, all of Southern Ontario is pretty much a disturbed landscape, or restored areas, or in the middle of a forest of urbanization. So there's a lot of invasion by this plant around there. And you can see there we're actually standing around the dog strangling vine, and then it goes into spreading under the trees as well. So it seems to shade tolerant as well. It was first introduced in Toronto around 1889. That's where the invasion epicenter is. It's very abundant in the Don Valley since the early 1900s. And today the species is pretty much a priority invader in Ontario. So some of the questions we asked were, have negative plant soil feedbacks. So as time goes by, are we seeing the legacy that this plant leaves in the soil and the soil biota that interacts with its roots? Does it become stronger? So will the plant experience negative feedback? So biomass declines over time. The other question we ask is whether or not there's accumulation of potential fungal pathogens over time in this plant. So to answer these questions, back in 2015, my first PhD student, now Dr. Nicola Day, and this month she actually was appointed professor at Victoria University in Wellington in New Zealand. And so Nicola went to the locations that are identified on this map, and she sampled DSV populations with different ages. So you see old, over 100 years old, between 50 and 60 years old, and then some younger populations. And then what we did then, we grew DSV in soils that were inoculated with microbes from the populations of different ages. But what we found, as you see here, is that contrary to our prediction, and even after 100 years of establishment, we found actual evidence for still an overall positive and not negative soil biota effect. When we used molecular tools to look for the potential accumulation of pathogens, we also found that DSV in young, intermediate, or old populations establish associations with bacteria and fungi known as pathogens in other species. But this didn't seem to change irrespective of the age of invasion, as you see here. So these negative results are important, though, because they highlight that even after 100 years, some invasive species continue to show no evidence of showing pathogen susceptibility, potentially population declines, and so they can, which begs the question, continues to beg the question for how long can different populations persist as invaders, and how can we best predict that? So in a subsequent study, we actually isolated potential pathogens from inside dog strangling vine. We plated them. You can see some of them here. And we were able to test plant growth responses, but they were only significant when a specific mixture was present. And this is in very artificial conditions in the lab. So, but the data did indicate that in principle, growth declines are possible. Now, fast forward to 2021, we just published this paper with my master student, Catherine Ducheneau, and with collaboration with Dr. Colati at Queen's University. Catherine is now a PhD student at Georgia Tech. And so we published this data where you can see, showing that soils from forests that are invaded by garlic mustard contained a greater richness of fungal pathogens, right? So here you can see fungal pathogens. We looked at other organisms as well, fungal animal pathogens and saprotroph richness, but where we found some evidence for pathogen accumulation was in populations that were in soils that were inside garlic mustard populations relative to the control adjacent sites that were outside the areas affected by the invasion. So some evidence for that these populations seem to serve as a resource for potential pathogenic organisms. Now, whether or not these pathogens are detrimental to the invaders or can spill over to the native population and aid in the invasion process, that still remains to be seen. But so in any case, though, the evidence is mounting that invasive plants, they do accumulate pathogens, and that through feedbacks with soil biota, their populations may start declining over time. So here on the left is a paper from 10 years ago showing that negative feedback. So in different plants that were established in New Zealand, so that negative feedback becomes more negative with residence time for those species. And on the right-hand side, we see data that was just published this month on garlic mustard. Again, this is from Bernd Bloss's lab, showing that garlic mustard populations, they start declining with time. And as you can see here, after a number of years, around 8 to 10 years, you start to really see it. The number of plots where garlic mustard is present is declined. They looked at these plots for in the long term, and you need to do that. So when you see these kinds of surveys that MNR does with that was just being presented, the importance of every year getting out there even in a pandemic, so that we get science going is extremely important. So here, this data shows that the data show that garlic mustard populations, they do start declining with time, and this is actually more pronounced in the northeast, where the invasion is older. So one question that we can then ask is tied to the relative importance of combined effects of multiple pathogens, which could lead to naturalization or to the susceptibility to a disease outbreak. So going back to that figure that I showed at the beginning of the invasive species curve, so we have been thinking, where do we go from here? And so our lab is working on a framework where we propose that looking at invasive species traits, and here we're thinking of in plants specifically, it might help us better predict pathogen susceptibility. I mean, we're thinking of plants, but it doesn't mean that this approach can be also developed for other types of organisms. Now, for example, we know that species that have low leaf mass area, they're more specific to susceptible to pathogen attack than species with a high leaf mass area. As you see, there's a lot more surface area for pathogens to potentially attack. So you could think of a leaf from a banana tree, for example, as opposed to a needle from a pine tree, and that's just one trait. So this could be important in the trajectory of the invasion over time. So one of the things we did was, so based on this actually, with Dr. Catherine Fahey, who's currently a postdoc in the lab, we used the data collected, so from the literature, data on pathogen impacts on invasive plants, and we applied a multivariate model to associate plant traits with pathogen effects, which you can see here on this plot, on this figure. We compiled trait data from the TRI database for each invader, and then we selected all the traits that were available for at least 70% of the species. Based with that information, we ran a principal component analysis, and then we calculated the pathogen effect as the relative difference in performance, so survival or reproductive output growth. So we measured that between the control and the pathogen infected plants, and we averaged that across all the available studies for each plant pathogen combination. So for studies that tested multi-pathogens on a single invader, only the most pathogenic was used in the analysis, because we are interested in that susceptibility to those pathogens that are actually able to cause disease, and not necessarily to an overall accumulation that may not have effects on the plant. So when we think about pathogen susceptibility, is actually to novel pathogens that are acquired in introduced range that leading to population level declines. So that's what we find here. You can see that there's actually 3 species in our data set that we could not find pathogen data available, and those are in yellow, but we were able to use some functions to assign missing data. So it's in terms of the model, it's in the process of being developed. We have this paper already out as an early view, so it's up for comment in Authoria, and so feel free to access it and comment as we keep developing this. So the factors that are going to be involved, we have quite a bit of data, but our proposal is that now we need more trait data, we need more experiments, and people on the ground looking at evidence for disease in invasive plants, so the model can become can improve. So the factors that are involved in future research directions for predicting pathogen susceptibility, will lead to model accuracy that is expected to increase as questions that are associated with plant traits are answered. Also, pathogen traits, the influence of context dependence is going to be important to take into consideration. And so also trait interactions with plant traits. But as we get more information on and data, both on trait data and pathogen susceptibility data, it will help refine this model. And once we have that previous principal component analysis with more data, then we can, based on all those traits, we can pinpoint, okay, what invasive plants may be more susceptible to disease first. Obviously, as you could see earlier on, dog strangling vine would not be. We have to look at the traits of that particular plant in relation to other traits to try to understand that. So that was the take-home message for today. And yeah, I'll take any questions. I'd like to just thank my funders and take any questions. Thank you.
Thank you so much. We will go into questions now. So if anybody has any questions, please feel free to raise your hand or write in the chat or the Q&A, and we will address those. I'm quickly just going to say that Taylor Scarr did put a little message to everybody in the chat here saying that CFS spraying of Gypcheck with the federal government doesn't carry out operational spraying, just research on the same subject. Silvar Technologies from Fredericton, New Brunswick, holds the license from the Canadian Forest Service to produce the product. Silvar has submitted a registration request for a liquid formulation. They hope to have it approved for this coming year, but that is in the hands of the Pest Management Regulatory Agency, and they would still have to produce usable quantities. So if anybody else has any questions, feel free to jump in here.
I have a couple of questions for Dr. Antunes. So first of all, you mentioned, that there's more information required to continue to refine the model that you are building to relate traits to disease susceptibility or to pathogen susceptibility. I'm wondering, is there like a public or citizen science component of this research that you're planning or, that you could see being useful? Do you have any tools in place that, could be distributed in that way?
Yeah, I see a major need for that. We need to have people on the ground and then detecting these diseases and looking at population level decline. So I see a major need for that. But we are putting this framework out there, and alongside with it, we have the spreadsheet on how we calculated these trait values and what traits are we considering and why. So the framework is there, the data that is available is there and how it was collected and tested, but we need a lot more. So it would be wonderful to have this component included in things like the databases that are used, EDD maps and things like that, when people are on the ground, sending pictures, if they can send pictures of potential diseases that are affecting these plants. And if we can even think about ways of having areas with permanent plots where citizen science could go and monitor these plots regularly and things of that sort, I think it would be wonderful. And we need to develop and think more about, because we've had, invasion science is fairly recent, and now we focused and continue to focus a lot on the alien species, which ones might become invasive. But now that we have all these invaders and we are aware of them and starting to accumulate data on these invasions, we can also start looking at the dynamics of pathogen accumulation, disease development, and use this in our predictive models. So to answer the question, yes, I see a major need, and maybe working with Invasive Species Center, we could come up with some better solutions for help in this process.
Absolutely, that's great. And then, sorry, just one more question that I had. So you mentioned that it was up for comment. Is that public comment or is that peer review? Like, is there a place that, you know, we could comment on that or
yeah, Authority is it's like bioarchives. The paper is actually in the processes of being reviewed, going through revision, and we're just doing revisions, so it will be published, I think, fairly soon. But we put it out anyway, so it's an Authority, and I'm happy to share it, and people can comment. And you know, it's the reviewers sent so many comments already that you know, we're improving it, and we think we can address all of them, which made us even more. It's fairly new. I think that's the part of, you know, as I mentioned, all these invasive species hypotheses are not centered here, and so it's fairly new. Also, working with pathogens, as everyone knows, is very difficult. What's a pathogen in one plant might not be a pathogen in the other plant. So we do need a lot of data, but we don't have it yet. So it's calling for data and providing a framework. But yes, to answer your question, I'm happy to share the paper, and then people can send comments.
That'd be excellent. Thank you.
Great. So we also have a question in the Q&A here for you. You mentioned the pathogens in the soil may actually benefit the invasive plant in its ploy to take over our natural areas. Is this the allelopathic effect that we hear about with regards to DSV?
Well, DSV is a plant that we know it's highly mycorrhizal. So I'm not surprised that it takes advantage of. In this, I'm hypothesizing, you know, there's enemy release is introduced, and what we see is that this plant continues to take advantage from the soil biota that it interacts with. So when it grows in sterile soil versus non-sterile soil, it grows a lot better. So it might be the novel mutualisms, the novel beneficial microbes, and the lack of enemies that is helping this plant. And this happens with quite a few plants early on in the invasion process. And then what we're trying to think about is what happens later on. Allelopathy is when with something that seems to be stronger with Alliaria petiolata, with garlic mustard, rather than with dog strangling vine, and that is the action of the plant suppressing other plants. And so that's not something we've studied in dog strangling vine, but we have looked at Alliaria petiolata, and what we find is that allelopathic effect on beneficial fungi doesn't seem to be as strong in the field as the literature portrayed it at first, because a lot of those experiments were done in the greenhouse. So, and now we're seeing these data also showing that over time, dog strangling vine seems to be declining. Garlic mustard, which also then starts to beg the question: how much should we prioritize this species for control? You know, if after 12 years it starts declining. So this is starting to change the way we think about the temporal dynamics of plant invasions.
Awesome. So there is a question to clarify: does DSV grow better in sterile soils than in non-sterile soils?
It grows better in non-sterile soils.
Okay. And then somebody's also asking if you can provide your contact info if they're interested in establishing PSPs for citizen science.
Oh, absolutely. You can, if you Google my name and Algoma University, you'll find my contact. It's pedro.antunes@algomau.ca. Feel free to send me messages, and I'm happy to answer them.
Good morning, everyone. My name is David Nisbet, and I'm the Partnership and Science Manager at the Invasive Species Center, coming to you from Sault Ste. Marie this morning. And I want to thank Forest Ontario and Natural Resources Canada for putting this session together and for the invitation to speak. I'm going to be talking a little bit about knowledge transfer today and the importance of connecting science to action and translating that science-based knowledge and information to broad audiences. If you're not familiar with the Invasive Species Center, we're a nonprofit organization based in Sault Ste. Marie, Ontario, that connects stakeholders' knowledge and technology to prevent and reduce the spread of invasive species that harm Canada's environment, economy, and society. We were formed in 2011, so 2021 is actually our 10-year anniversary. So stay tuned for lots of exciting news and announcements coming up soon about some of our 10-year anniversary celebrations. At the ISC, we see our work as being divided into 3 priority areas. The first one is catalyzing action. So working with a broad range of partners to catalyze action on invasive species prevention and management through creating networks, contributing to response and management efforts where we can. We're also working a lot to increase and diversify investments in invasive species. So that's trying to make more funding available on the landscape for everyone that manages invasive species and come up with new and innovative funding mechanisms to try and facilitate that action. And then finally, what we'll be focusing on most today is our efforts to share knowledge. We work to increase and share knowledge from the knowledge generators to the knowledge users and try and create more awareness and action on invasive species. So in case there's anyone on the line that's not currently convinced about why invasive species are bad, I thought I'd just start my presentation by throwing up a list of all of the reasons why we don't like them. In forest systems, they kill trees first and foremost. It creates dead and dying trees on public and private lands. And there's also implications for climate change. As trees die, there's less carbon storage. It could increase risks of flooding. It can increase energy costs, diminish property values, permanently alter landscapes and ecosystems, impact human health, and the list goes on and on and on. So I hope everyone's with me in agreement that invasive species are bad and we don't want them in our forests. When we look at how invasive species are spread, a lot of the common pathways include things like movement of packaging materials like wooden crates, hitchhiking and traveler's luggage, moving from place to place in raw untreated firewood. Could be seeds or microorganisms within topsoil and garden materials, attachments on nursery stock, seeds and things stuck in hiking boots or recreational or commercial equipment, roadside equipment, clothing, pets, and the list goes on and on and on. So to summarize, really, pathways of spread for invasive species are primarily people, and not just stock photo models, but everyone, probably people and maybe some of us on the line today have been guilty at spreading invasive species at one point or another. So when we're looking to prevent the introduction and establishment of invasive species, we really need to keep that human element first and foremost and focus on addressing human-facilitated pathways of invasive species movement. When we think about knowledge transfer, we think a lot about outreach and education, but really knowledge transfer is important throughout the whole process of invasive species prevention and management. It could be, communicating new regulations and legislation in quarantine areas. It could be taking the information from the research and turning it into a management practice for a practitioner or a land manager. Or it could just be public education and awareness. So throughout the whole process of invasive species prevention and management, a focus on communications and knowledge transfer is important. Pedro showed the invasion curve, and I'm going to show it again. It really demonstrates that the sooner you can catch an invasive species and the sooner you can prevent it or eradicate it, the greater the long-term benefits will be. It's really that old saying that an ounce of prevention is worth a pound of cure. And if you look at the bottom axis of the chart here, it shows the economic returns. And this is a generalized model and it's different depending on the species or the situation. But in general, we think for every $1 invested in invasive species prevention, you achieve $100 saved of long-term costs that you'd otherwise be faced with. So if we look at this in terms of species, something like the emerald ash borer, it's at the far end of the curve where we're in that long-term control phase where costs are high and we're really, being forced to spend lots of money on an annual basis to control emerald ash borer and mitigate health and safety impacts and try and manage our forests. Where we want to be, though, is on the other end of the curve, where we look at invasive species that haven't yet crossed our borders or come into Canada, or even on a smaller scale, come into your local municipality or the area that you're managing. You really want to be on that end of the curve and look at what species are coming next. And that's where, at the Invasive Species Center, where we prioritize our knowledge transfer measures. So the colorful insect I've put down there is the spotted lanternfly, and I'm going to be talking a little bit more about that today. But just to make the point that it has not been introduced into Ontario or Canada, and prevention is still possible. So the spotted lanternfly, I'll tell you a little bit about it. And if you look at the picture on the left side of the screen, that's actually a whole swarm of the lanternflies on an apple branch. It's a leafhopper insect that was first detected in North America in 2014 in Pennsylvania. It feeds in swarms, as we can see from the photo, attacking more than 70 species of trees and plants. And it's especially a threat to grapevines. It seems to really prefer and attack grapevines. So it is a big risk for the wine and fruit industries where it's introduced. Beyond grapevines, it also has been found feeding on species like black walnut, maples, and oaks, and it preferentially feeds on tree of heaven, which is an invasive tree species. It hasn't really been shown to kill or have long-term impacts on some of the tree species, but it does create aesthetic issues because it secretes a honeydew that creates mold growth all over the base of the trees. If it's a backyard tree, it will rain down the honeydew on your lawn furniture and, not make a great backyard friend. So there's big socioeconomic impacts and aesthetic concerns beyond just the economic concerns for the wine and grape industries. I mentioned it has not been detected in Canada, but it has been intercepted as far north as New York State in areas that border the Niagara region in Ontario. It also spreads through a number of mechanisms. It lays egg masses that look quite similar to gypsy moth egg masses, just a little bit less fuzzy. And it lays these egg masses on essentially any outdoor material it can find. Commonly, they're found on things like lawn furniture, vehicles, landscape materials like stones, nursery stock, and other commodities. You can see in the photos that it also is an avid hitchhiker, and it'll latch right onto your car or your clothing and can go for a ride with wherever you're going to take it. So this is a priority for outreach and education and knowledge transfer, and we really need to get the word out there. But before we do that, it's always good to take a strategic approach. We don't just jump in with creating fancy videos and graphics right away. Really need to think about why we're communicating and what results we want to achieve. When you're doing any sort of knowledge transfer, thinking about your target audience is very important to make sure that you're communicating to the right people with the right message to achieve the desired result. And once you've identified those factors, only then you can start to initiate your outreach tactics. So for the spotted lanternfly, the why is that it poses huge threats to Ontario and Canada, particularly the economy of the wine and grape industries. The result that we want to achieve through spotted lanternfly outreach is to raise public awareness. It's a really flashy, showy insect, and we need more people on the ground looking for this bug and reporting any sightings. Our target audience for this, I've mentioned a few times that it impacts the wine industry and the fruit industry. So we want to get the attention of people in that sector. Also arborists, municipalities, landowners, people that have a close eye on the forest and would be able to recognize something like this. Looking at the climate models for spotted lanternfly projected spread, it shows that areas of southern Ontario are at highest risk, especially corresponding with where tree of heaven is known to be present. So we can actually target our outreach very specifically at those regions of the province where we see the highest risk for spotted lanternfly introduction and establishment. And through the next series of slides, I'm going to talk about the how. So now that we know who we want to reach and why we want to reach them, only then can we decide, how are we going to do it. And the first initiative is really looking at partnerships and collaborations. I mean, the Invasive Species Center has a great network of partners and stakeholders. But working with other partners and leveraging other networks, the message can go even further. So when we're starting to think about knowledge transfer for this insect, we put together a group of collaborators that could help us achieve our outreach goals. So we've engaged members of the apple growers of Ontario, the grape growers, the tender fruit growers. We have some of our government partners at the Ministry of Natural Resources and Forestry and MacQuarrie, some federal government partners at the Canadian Food Inspection Agency, and other non-government partners at OFAH and Eastern Ontario Model Forest. So this is our education and outreach committee, and we meet on a regular basis to talk about how we can most effectively reach our target audiences through our combined efforts. We also created a home for all of the spotted lanternfly information on a species profile on the Invasive Species Center website. So this is an area where we can put the common messages that other partners can use for outreach. We can upload all of the resources that we collaboratively develop and share resources that others have developed. This page has the ability to be updated in an instant. So as things like distributions or regulations or new research emerge, we can quickly update the page and make sure it's current. We have also produced digital and physical resources in collaboration with our partners. We developed a new spotted lanternfly poster and also a technical fact sheet. And we have stacks of these in the office. If anybody wants them, please just contact me and we'd be happy to send you a stack of them. I know recently digital outreach has been more doable than in-person outreach, so these are also available for download from the website. We host digital learning webinars where we've invited speakers from Pennsylvania to come and talk about their experiences, the research that's going on down there. So landowners and managers in Ontario can learn about the lessons from Pennsylvania. Since it's such a flashy and showy insect, it really is a great candidate for social media. So here's some examples of Instagram ads and Facebook posts that the ISC has done. And just a quick note that on social media, it's really easy to boost your posts or pay for promoting your posts. And that small amount spent to actually put some money behind what you're posting goes a long way in increasing your reach. It's really worth your time and effort if you want to increase the engagements and views on the content that you're sharing. Even beyond the members of our education outreach committee, we reach out to other partners and see if we can integrate spotted lanternfly content into their platforms and into their communications networks. Here's just a few examples of some different newsletters and articles that we've developed the content for, but posted through some of our partner platforms. So we've worked with the Biodiversity Education and Awareness Network, the International Society of Arboriculture Ontario chapter, and also Ontario Parks. Can't forget about traditional media. A lot of the target audience, might not be on social media and might not read the newsletters of our partner organizations. So some of the larger publications are always looking for great content on invasive species, especially those charismatic ones like spotted lanternfly that you see it and you say, what is that? It's easy to get the attention of the media and the broader public. If all else fails reaching your target audience, sometimes the best effort is to go directly to them. And this is some of our team at the Ontario Fruit and Vegetable Convention, educating some winery owners on the risks of spotted lanternfly and some of the identification and detection information. These days, conferences in person aren't as popular or aren't happening at all. So we've switched a lot to digital outreach as well and digital calls to action. A few months ago, we ran a campaign where we were inviting members of the public to report sightings of their tree of heaven, which I mentioned is an invasive tree species that is also the preferred host of spotted lanternfly. And we ran a campaign getting people to report sightings of where their tree of heaven are. So as spotted lanternfly get closer and closer to Canada, knowing where the tree of heaven exists already is very helpful in determining what areas might be highest at risk and where we should focus future monitoring or outreach efforts. And I want to end with a bit of a story of a report of spotted lanternfly this past summer to just sort of demonstrate the power of social media and rapid communications in the digital era. I'll preface this by saying it was false, so I don't want anyone going away today saying spotted lanternflies in Ontario, but I'll take you through what happened. So this summer, a member of the Reddit community had posted this photo of what are clearly spotted lanternfly nymphs on an entomology forum, asking the other users, what is this bug? I've seen this in Mississauga, Ontario. I don't know what it is. There was a very quick response, again, because I think spotted lanternfly is so charismatic and easily recognizable that the message exploded. So it was all over Twitter, especially through the entomology communities. Emails were popping up. My inbox was full of people talking about spotted lanternfly and how it was in Ontario. People at Natural Resources Canada, the Ministry of Natural Resources and Forestry, OMAFRA, the Canadian Food Inspection Agency, and others were aware of this report within 24 hours of when it had been posted on Reddit. And action was initiated. Government officials were following up with the person who posted this picture to try and confirm if it was in fact legitimate or not. Turns out it wasn't. The person who posted this was, I guess, sent the photo by his mom, and his mom didn't say where the photo came from, just said, hey, do you know what this bug is? And he posted it to Reddit and wasn't actually aware that the photo was taken in New Jersey and not in Ontario at all. So it turns out that thankfully it was a false report. But even though, it was a bit of a wild day, it really does demonstrate the power of some of the communications networks that are in place. It's amazing to see that within 24 hours of this being posted in an online forum, there was such response from all of the appropriate government partners. It really demonstrates that social media is a super powerful tool for identification and detection of invasive species. I guess Reddit is the same community who can break the stock market, but they can also help identify and report invasive species. So there's good and bad to online discussion forums. Once the report was out, action was initiated right away to confirm if this was legitimate or not. And it really demonstrated that there was great public and private interest in this situation. You know, CBC Radio called us and wanted to talk about the spotted lanternfly in Mississauga. So through multiple channels of communication, the word really got out there. And I'd say that the best part of this situation for me was just looking at some of the comments that were posted on Reddit following the initial picture that was posted. These are just other Reddit users, and if you look at all of their comments, and the list goes on, I've just pulled out the top 3, but there were many more comments after this. Every single person was saying, you need to report the sighting. This looks like spotted lanternfly. You know, contact the Ministry of Natural Resources and Forestry, contact your agricultural authority. It was really amazing to see that community of people that were engaged in, you know, helping to stop this threat of invasive species. So that really tells me that the message has been heard, and all of the knowledge transfer work that not only the ISC is doing, but all of our partners have been doing is really serving the purpose that it's intended to do. It's creating this engaged community of citizens and community members who are taking part in invasive species reporting. So just to finish off my presentation today, just a few final thoughts, takeaways and tips when you're thinking about communicating about forest pests. As I mentioned, always define your objectives and strategy first, and tailor your messages to the specific platform and target audience you're trying to reach. Keep in mind species biology, things like geography, current distribution and projected future distribution can really help you target in the people that you want to reach with this specific information. There's lots of information on risk assessments and climate modeling, and all of these science-based pieces of information should be integrated into your communications approach. Species life cycle is also very important. If you're talking about, you know, something like Hemlock woolly Adelgid, and you're telling people to look for the woolly masses, that message is much more effective if you communicate it at the time of year when the woolly masses are actually visible, and not the time of year when they're not visible. So you need to pay attention to time of year, species life cycle, and all of the other biological factors. It's also easy to focus on, you know, simple identification features and not get too technical with the identification information. You know, you want to give the broader public the basics that they need to know, what to look for and how to report it. I always think that a false report or a misidentification is much better than no report or no identification at all. I mentioned the importance of talking with partners and collaborating to leverage collective networks and really continue to amplify the message. We always try and include a call to action in any information on how the audience can help. Invasive species messages tend to be, you know, sort of doom and gloom a lot of the time. And we have an audience of people who want to help and who want to contribute. So including a call to action, like, you know, report your tree of heaven, really lets people know that they can help and they can contribute, and there is hope in invasive species management. There's many social media platforms out there that users are on, and it's really easy to make use of them. Through simple social media marketing, engaging in online discussion forums, looking for existing online communities of the audience you're trying to reach are a great way to become part of the conversation and spread the message you're trying to get out. And also, I know it's you know, hard to think about now in the age of COVID, but really don't forget about the value of in-person communications. Digital communications go a long way, but you know, there's nothing like having a conversation one-on-one at a conference or meeting a woodlot owner in person to talk about risks and actions they can take. And finally, the point that I think is most important, which is why I bolded it here, is that everyone in the science, research, and natural resource management community can and should actively engage in science communications. You don't have to work through an organization like the Invasive Species Center. You can take action yourself and start to communicate your research findings out to broad audiences. You know, unless your name is Donald Trump, I think you can easily create a Twitter account and start to get your message out there and let people know about the work that you're doing and what it means for them. Taylor mentioned it this morning, but you know, in the digital era and the age of fake news and conspiracy theories, we really need more science-based content out there. And the more people we have spreading that message, the better it will be. And finally, I just wanted to mention that if you'd like to learn more about the Invasive Species Center and stay up to date with everything that we're doing, feel free to visit our website. You can sign up for our newsletter. We put out a bi-weekly media scan. We send out event and webinar invitations and much more. So visit our website if you'd like more information. And finally, I just wanted to thank everyone for your time and let you know that if you need help with science communications and you want to get your message out there, but you're not sure how, please contact the Invasive Species Center and we can help.
So thanks everybody for attending my talk today, and thanks Forest Ontario for hosting us and having us being a part of the morning. I am going to talk to you today about some work that we're doing on a novel control of buckthorn on the landscape. And I wanted to begin the presentation just by first really quickly introducing Lallemand. Lallemand, oops, sorry, I just advanced by accident. Lallemand is a Canadian company founded in Montreal at the end of the 19th century. And it's been owned by the Chagnon family since 1952. Just looking currently, we have about 4,000 employees in 45 countries with a number of different plants and research labs and offices around the world. So Lallemand is a global leader in the development, production, and marketing of microorganisms and their derivatives. And within the company, as you can see in the slide here, there's several different business units, including the one which I am part of, which is the plant care business unit. And many of you on the call today may be familiar with bioforest, perhaps for the product treason, for emerald ash borer management. And just so you know how the two fit together is that bioforest became a subsidiary of Lallemand Plant Care in 2015. So at Lallemand, a plant is really viewed as an ecosystem in which microorganisms play an essential role. And so here you can see that we have a whole line of products which are suited to various elements of the plant itself and to plant health. And the one that I'm here to talk to you about today and the research that we've conducted is not yet on this list as it's just recently been added to our portfolio, but I'm going to get into that just now. And so this brings me to the topic of today, which is one of our current initiatives in forestry, which is the use of louside chondro as an active ingredient for invasive buckthorn control. So just to highlight the problem itself when we're talking about forest health, looking to buckthorn, both common and glossy buckthorn are invasive to North American forests. And some of the issues with buckthorn on the landscape are its ability to outcompete natural regeneration, altering the ecosystem for both flora and fauna. It can form these super dense monospecific thickets as seen in the photo here. It can even alter the soil, among many other issues. And it's also incredibly difficult to manage. So it has this ability to produce prolific sprouts post-harvesting from the stump, which over a course of a year or two can really grow substantially in height. It also can have significant seed bank in the soil, which can remain viable for many years. And both common and glossy buckthorn can be found in both urban and rural landscapes. And so, you know, it can be quite prevalent. And so at Lallemand Plant Care, we've been exploring the use of a biological herbicide as a tool to help control this invasive plant. So without further ado, that's what I will get onto now. So looking at the fungus that was selected to battle this invasive plant, Chondrosterium purpureum was selected due to its history, its mode of action, and its development as a biological herbicide. And so Chondrosterium purpureum is a naturally occurring wood decay fungus, which is present throughout Canada, and it can also be found globally. It has a really long history of being recognized as a colonizer of stumps of freshly cut trees and as a wound parasite. And looking to its mode of action, Chondrosterium purpureum is a hemibiotrope, meaning it infects living tissue, eventually killing it, and then it can continue to develop on dead tissue in competition with other saprotrophs. And the fungus is often called a silverleaf fungus, which is due to the secondary mode of action, which targets those stump sprouts that I had mentioned earlier post-harvesting. And so this is where the Chondrosterium purpureum actually releases an enzyme, which then disrupts the middle lamella between the epidermal cells and the palisade cells of the leaves in those shoots, causing the two layers to separate. And so it's this change in cell arrangement which results in the change reflectivity of the leaf, which causes that silver appearance, which is what we call silverleaf disease. And so it's this decline in those post-harvest sprouts which typically lead to shoot mortality. And it's these two modes of action working together to colonize the stump and control those sprouts post-harvest, which results in overall efficacy of stem mortality. So a common question that we get surrounding the product itself is the risk to non-target species. And so I wanted to just spend a few minutes here to address that question. And so as I mentioned previously, the fungus is naturally occurring here in Ontario and across ecosystems in Canada. And in order to colonize something, it requires a fresh wound. And so in terms of the product itself, it is a concentrated targeted application, which is applied directly to the cut stump surface or to the girdling wound. And it's further targeted due to the fact that the product is a paste, and so it's not airborne in its application. And so the risk to non-targets is incredibly low, as operators would only be wounding and directly applying the product to the buckthorn themselves. And additionally, Chondrosterium purpureum has actually been studied for quite some time for its potential for use as a biological herbicide. And there are several literature papers which assess its use as a commercial product and the risk to our forests. And so this snippet here that I'm showing on the slide is a paper which actually compares the use of the product and the resulting spore load compared to that which is naturally present already in the forest. And it determined that it's of the same magnitude. So meaning that by using Chondrosterium purpureum as a product and adding it into the system did not increase the Chondrosterium purpureum pressure more than what is naturally occurring. So moving along then to its development as a bioherbicide, in 2009, Lallemand chondro with the active ingredient of Chondrosterium purpureum was registered in Canada to prevent the resprouting and regrowth from cut stumps of alders. That includes red alder, thin leaf, and Sitka, trembling aspen, red oak, ironwood, white birch, sugar maple, and chokecherry. And then the product, since 2015, Lallemand Plant Care has been exploring the potential of the product as a biological herbicide for the control of invasive buckthorn. And so in order to do that, we installed several research trials in Ontario and Quebec on both common and glossy buckthorn. And so I'm going to go through some of those research trials now with you. And I first wanted to outline the protocol that we followed for these research trials. And so we selected two management options to test. We were looking at cut stump operations and girdling the stems. And so in both operations, we always, every stem was either cut or girdled, depending on the operation, of course, that we were using. And then a certain number of stems in each one of our plots were selected for long-term efficacy. So you can see in the photo there, we would spray paint the bottoms blue and attach a metal tree tag so that we could find them at all future assessments when we went back to the sites to evaluate efficacy. And what I mean by going back to assess the efficacy is that due to the nature of louside chondro being a biological product, it takes time for the efficacy to be fully expressed because the fungus has to colonize the stump. So the trials were monitored for up to 24 months harvest and application. So some of the results that I have to share with you, I'm going to go through those now. And so here we can see a trial from Simcoe County, which was a cut stump operation, which was assessed at 12 months post-harvest and application, as well as 16 months post-harvest and application. And by that 16-month timeline, we were recording 89% mortality of the stumps that were cut and treated with louside chondro, compared to those that were cut with no product applied. And so examples of the dead stumps there can be seen in the photos on the left at the 24-month timeline. So you can see that stumps, that sprouts were produced, but by 24 months, the whole system was considered dead. And so here you can see what some of the sites that we were working in looked like. This is another cut stump trial that we worked in the Lallemand region of Quebec. And in the top photo there is the trial site immediately post-harvest. So you can see the density of the buckthorn under the red pine stand from the surrounding edges of the trial that weren't harvested. And in the bottom photos was the final assessment at the 24-month harvest timeline. And so that's actually me there on the left in a plot treated with louside chondro at the 24 months post-harvest treatment compared to the control plot on the right-hand side. So quite a visual difference between the two plots with treated, cut and treated, and then cut with no product applied to the stump top. So this is now another cut stump research trial that we established also in the Lallemand region of Quebec. And I just wanted to show you another example of some of the field observations that we saw. And so at just 3 months post-harvesting and treatment, here you can see here difference in the sprouts from a control stump versus those from a stump that were cut and treated with louside chondro. So the shoots on the treated stem are much shorter, they're smaller, they're chlorotic, they're generally unhealthy looking. And so already an early sign that the fungus is colonizing the stump and doing what we are hoping for. And so that trend continued at the 12-month assessment with really vigorous sprouts coming from the control stump, as you can see in the photo on the right there, compared to one that was cut and treated with louside chondro. And at the time of the 12-month assessment, we would have still called that stump that was cut and treated alive because it still had some foliage, as you can see just in front of my clipboard there at that time. But if we look at the data for the 24-month assessment, that stump would have succumbed to the fungus, and we would have called it dead because this site had 100% mortality by that 24-month timeline. And so at both the 12 and 24-month assessments, there was statistically greater mortality of the treated stumps compared to the untreated controls, as you can see in the figure here. So as I mentioned, we also conducted research trials with girdling stems plus louside chondro as certain instances that type of operation is preferred. And so in the graph here, we can see that at each assessment timing, the stems that were girdled plus having louside chondro applied to that girdling wound has statistically significant greater mortality in treated stems at all 3 assessment timings. Compared to the untreated controls. And so finally, by that 24-month timeline, we observed 93% efficacy or mortality of the stems that were girdled and treated with louside chondro compared to the untreated controls that were girdled only. And this trial itself took place in collaboration with the town of Oakville. And so the last one that I wanted to show you today was actually established in the city of Montreal in 2019. It was another trial where the girdling method was used. And so I wanted to just show you in this photo here that you can see what a control plot would have looked like in the photo on the left. So the stems were girdled and left as is. And then on the right is an up close picture of a stem that was girdled with Lallemand chondro applied to that girdling wound. So the site was visited 4 months post-treatment, and in the photo on the top right there, you can see an overview of one of the control plots with really healthy looking shoots, where on the bottom left and in the photo above it are plots that were treated with Lallemand chondro. So it's a little bit tricky to tell with the foliage that was on the ground there, but overall very poor looking shoots, some shoots that were dead already. And in the photo on the top, you can see that was an example of one stem that was very far along with the shoots already exhibiting silver leaf disease. And so at the 16-month assessment timing, many of the poor looking shoots previously observed were then considered dead, as you can see in the photo examples here. And in some cases, the fruiting bodies of the Chondrosterium purpureum were observed as well. And if we just briefly look at the data there, the 16-month assessment time that the stems that were girdled and treated with Lallemand chondro had 72% efficacy compared to the controls at 33% efficacy. And we'll follow up with this site to get that full 24-month data still. So this one is still a work in progress. But just a brief summary then of the work and examples that I had just gone through. These are the completed trials, except for the last one there. You can see the city of Montreal, where that one's still ongoing. But the other ones are all completed with efficacy consistently from 81% to 100% between the different trials that we've completed. And so I just wanted to briefly touch on a few notes about the product itself in case you were interested about that. And it's a manual one-time application through a 1 liter squeeze bottle. And one thing to note is it is important to keep your stump tops or girdling wounds low. And the reason for this is just simply because the higher it is, the more the fungus has to colonize. So we recommend keeping them around that 10 centimeter mark above the ground. And for timing, it's really important that the product be applied when buckthorn is completely leafed out. It's usually from mid-June to mid-July, depending on climatic conditions, but it would depend on your area. And just the reason for that is that it's the time when the buckthorn are the most vulnerable. And also a note about temperature. The product needs to be kept cool. So it could be stored in a fridge until you're ready to use it and then simply brought out to the field in coolers. And I also just wanted to note that as far as temperature of the site that you're treating, it's important to have some element of shade if it's possible. And the shading could be simply, if you're girdling, it could be the stem itself that's shading your wound. But the reason is you want to help incubate that fungus to help, optimize the conditions for establishment. So you could have a desired overstory species where, in some of the examples I showed, you're keeping that desired overstory and you're trying to control the buckthorn in the understory, and that could provide enough shade. And another benefit to providing some shade is just preventing a sudden exposure of full-on heat to the forest floor, which helps to mitigate that seed bed that's sitting in the soil. You're just helping to prevent that full sunlight from causing those seeds to germinate. And so just a few further notes on some feedback about the product application. As I've mentioned, it is a biological solution for controlling resprouting and regrowth. There's relatively minimal PPE required. It's a one-time application and it's relatively easy to apply. And the application is very targeted, creating minimal risk to non-targets. So in summary, of course, we recognize that the management of buckthorn is incredibly difficult and often cannot be achieved through a single intervention. It requires several years of intensive management on the landscape. And we feel that the results of our trials really demonstrate that low-side chondro can be an additional tool to aid in buckthorn management, and it could provide a biological solution to managers who may be limited with chemical herbicide use, both from a regulatory or a public perspective. And additionally, it is a naturally occurring organism found in the ecosystems of Canada. And so lastly, I'm very pleased to say now that as of September 2020, the Canadian label was amended to include the use against buckthorn in addition to the already labeled species, which were those alder, the red thin leaf and Sitka, trembling aspen, red oak, ironwood, white birch, sugar maple, and chokecherry. And so with that, the product is now available for pre-order for this coming season. And of course, we will continue to work on innovative research and development to create solutions in the plant care industry. So that is all that I had for you today. If you have any questions, of course, we'll have the question session coming up. But if there's anything additional, we'd be happy to hear from you at the website provided here. And you can also check us out online as well. And a thank you, of course, to all of our collaborators for the trials that we worked with, including Simcoe County, Lestrie region of Quebec, the city of Montreal, and the town of Oakville, and of course, our team at Lallemand Plant Care. And that is all I have for you today.
Thank you, Allison. We're going to go into the question period now, and we do have a few questions for you here. Okay. So does the fungus migrate from the host once introduced?
Okay, so just a second. Sorry, okay. I just didn't realize I had stopped sharing my screen, but I've got it. Does the fungus migrate to a host once it's been introduced? Was that the question? Yeah.
Okay. Yeah. Does it migrate from the host once introduced?
Right. Yeah. So basically, the fungus, once it's applied, like I mentioned, is very targeted to that application, and it doesn't translocate. So it's on that stem. There is the potential down the road that it could develop fruiting bodies and perhaps spread that way through spores, but it does require a fresh wound to whatever could be the non-target that it may infect. And so in order for it to affect something else, it requires a fresh wound. And there's also the note that the fungus is already naturally occurring in the ecosystems as well.
Thank you. Following the previous question, once the fruiting bodies appear, how contagious is the fungus to contiguous trees?
Okay, so that's kind of along the same question then. So if we do get fruiting bodies, which isn't always the case, but sometimes certainly happens, as we saw in the pictures there, then the non-target would require a fresh wound for that to be able to colonize a subsequent non-target.
Have you tested efficacy in fall and winter applications?
So we found through our trial work that the spring is ideal. And the reason for that is because at that time, the buckthorn has completely exhausted its root reserves. It's put all its energy into leafing out. And so when we create that wound and apply the chondro, the low-side chondro product to it, it is the most vulnerable at that time, allowing the fungus to get established, having the best efficacy at that time. So we recommend that spring, mid-June to mid-July timing for that reason, to optimize efficacy.
Awesome. I don't currently see any other questions in here. There was one comment sent during David's presentation saying spotted lanternfly has actually caused widespread mortality in grapes. Just to mention, it is believed to severely weaken heavily infested trees as well. It is more than just an aesthetic issue for them. After Ailanthus, the Acer genius is the next most preferred native host.
Thanks everyone for tuning in today. I was just sort of thinking about, it was sort of this time last year that Victoria was in the process of organizing the in-person event. It was supposed to be at Algoma University, and I remember at the time it was supposed to be, we thought it would be quite novel that we were going to have a part of it be an online component so that folks could join remotely. And unfortunately, we weren't able to run the event. And of course, folks know how things have developed in the last year. So it's wonderful to be here with you all virtually for this session. And thanks to everyone for putting it together. This has been really informative this morning, and I hope to contribute. So I'm going to talk a little bit about Emerald Ash Borer in 2020, some of the updates and the stuff we've been working on, and then some of the opportunities for management. So this is some of the stuff that we've been doing sort of with my group at Great Lakes Forestry Center in Sault Ste. Marie, and then I'll talk about some of the work my colleagues have been doing in other parts of in other labs at Great Lakes, and then some of the other work across the country. So. There we go. I'm going to just outline the talk today. I'm going to talk a little bit about where EAB is now, update on our biological control program, some recent work on ecology detection and control tactics, and then some work on ash seed collection, and then a bit about the deregulation process in the States. So this is the currently regulated area for emerald ash borer in Canada. So, folks in Ontario, we're sort of familiar with everything that the yellow area is basically the area regulated by CFIA for EAB, and it's sort of south of the line about Montreal River Harbor for those in Northern Ontario across to the Quebec border. Everything south of there is regulated, and that roughly corresponds to the expected range, to the known range of the insect in Ontario. So we know it's at far north of Sault Ste. Marie. We know it's in Sudbury. In Quebec, it extends a bit further north, and then we have the sort of what we call the satellite, and then off into New Brunswick. And the biggest, there's a couple satellite populations, one over in Winnipeg, another one in Thunder Bay, and then the one in Halifax. And probably the biggest change in this regulated area in 2020 has been the addition of New Brunswick. There was the find a couple years ago up in the Madawaska region, up in northwestern New Brunswick. And then more recently, we've had finds around Fredericton and then Moncton, and so they've basically regulated all the counties along the Trans-Canada Highway through New Brunswick. So that's this sort of band of yellow that runs through the province, and that's been the biggest change. These other areas, especially in Southern Ontario, have been regulated for quite some time. So I'll jump right in and talk a little bit about our biological control program. And folks have seen this talk before by myself or by others who might be familiar with what we've been doing for the past couple of years. And this is work that we do with a lot of partners. It's not work we can do without a lot of support, both sort of in kind and financial, from all of these folks who participated in the program. And most of these folks, most of these organizations and communities have contributed release sites for us throughout Eastern Canada. And I always like to talk about the folks that are involved in this project, the team. This work was started by Dr. Barry Lyons, who's now retired, enjoying his retirement on St. Joe's. And after Dr. Lyons retired, Krista Ryle picked it up, Dr. Krista Ryle. And then I've been looking after it for the past couple of years. But the person who actually does most of the work, all of the work, is Gene Jones here, the lead technician on the project. Gene's responsible for finding release sites, organizing releases with partners, and getting the material imported from the US. And then these folks here in the insect production unit at Great Lakes Forestry Center are the ones who grow a lot of the insects, and especially this year they did a lot of work so that we could have parasitoids for release in Canada, and I'll get to that in a little bit. And then we have our collaborators in Quebec and our collaborators in Nova Scotia, in New Brunswick, who take care of the releases out east for us. So in Canada, we've introduced 3 parasitoids: Ubius agrilli, which is the one here on the very small one on the left-hand side of your screen. That's the egg parasitoid, first released in 2009 in the States, and then 2015 in Canada. The one in the middle here is Spathius gallinae. It's from the Russian Far East. It was approved in spring of 2017, first releases shortly after. And this one matches our Canadian climate a lot better. It seems to do better in emerald ash borer than a 2-year life cycle, which we think happens more commonly in Canada. And then we have Tetrasticus plantapenisi, which is a larval parasitoid, and it's been released in 2009 in US and released in Canada since 2013. And this is kind of the workhorse of the biological control program in Canada. It's been the subject of the most releases, and it's certainly the most commonly released agent in North America at this point. So talk about where we've done our releases. So I'll just show you a series of maps. These are all the same, showing the release sites across Canada. up on the left here, this is the map for Ubius agrilli, and we're just showing here in this chart on the left the number of parasitoids that have been released each year. And you'll notice for the next couple of slides, these all stop at 2019, and there's a reason for that. So in 2019 we added these new release sites in brown, but these dots are all colored by the year of the first release set of sites. So what you'll see with all of these parasitoids is our selection of release sites and where we've been letting parasitoids go has sort of followed the invasion wave of emerald ash borer. So emerald ash borer was first detected down around Windsor, in Detroit, in the early 2000s, and has been spreading sort of towards the northeast in Canada. And so that's where we've been doing releases for all 3 parasitoid species. And so we added these sites sort of at the leading edge in Ontario, and then in Quebec, and then up in New Brunswick for Ubius in 2019. For Spatheus, we haven't been releasing Spatheus in Canada for very long. Our first release was in 2017, just one site up near Ottawa. We added a lot of sites in 2018, and then quite a few more sites in 2019 with the goal of expanding the program then. Again, following that wave of emerald ash borer in eastern Canada. And then finally, Tetrastigus we've been releasing in Canada for the longest, started down in 2013, way down in the southwestern part of Ontario. And then we've been sort of moving again up towards the northeast. And in 2019, we added these sites up in sort of North Parry Sound, and then east of Toronto, and then in Quebec, and then up in New Brunswick. And so our plan for 2020 was to expand our releases sort of as we've done every year, and finish off some of these sites that we'd started in 2019. So we'd had releases planned in Ontario, Quebec, and New Brunswick. And as with everything in 2020, we were impacted by COVID. So the majority of the parasitoids that we get for the release program in Canada come from the US production facility in Brighton, Michigan. And we get these parasitoids by Tim Ladd or Gene Jones will leave Sault Ste. Marie, drive down to Brighton, pick them up, and then bring them back, hand carry them across into Canada and then they ship them out to our partners who actually make the releases. And the folks will be well aware that we weren't actually allowed to travel into the US this year to collect those parasitoids. So that meant we had a much reduced capacity. We were only able to rely on the releases that were made, the insects that we grow at the insect production facility in Sault Ste. Marie, and we grow enough there, enough Tetrastigus and enough Ubius to supply about 2 or 3 release sites in Canada every year. And so what we did then is we weren't able to supply any release sites in Ontario because we weren't able to be able to coordinate the releases with partners. And so we sent the parasitoids out to Quebec and New Brunswick because we had folks that were able to make the releases, and that was only later on in the summer when we had access to collaborators who were actually able to actually do the travel that was necessary. Unfortunately, we weren't also able to make any releases of Spatheus gallinae anywhere in Canada because those parasitoids all come from the US. We don't have a colony in Canada. And so what that meant is our sort of Ontario sites didn't receive an allocation of parasitoids this year. So 2020 all wasn't bad news. What we did have very late in the year was some evidence of successful overwintering and dispersal within Canada at our very northern sites. So what I'm showing here is a map of the Algoma region and the very northwestern tip of the Upper Peninsula of Michigan. And in late November, Tim Ladd, who's the lead rearing technician at the program at Great Lakes, he collects small ash bolts to get emerald ash borer for some of our rearing programs. And when he brought these little ash bolts into colony in the building and warmed them up, instead of nothing coming out, which is what usually happens, we put the EAB into these. Tim actually had Tetrastigus emerging from these little mini bolts of ash. And then again, later on in January, he had the same thing happen with some bolts that he actually collected right in Sault Ste. Marie. And so what this tells us is that these are naturalized populations of Tetrastigus that are establishing and reproducing in Algoma. So both at this site south of Echo Bay and then again in the Sioux. Now, the next question is, of course, where do these parasitoids come from? So our 2 closest release sites are one that we established in 2017 at Fort St. Joseph on the very southern tip of St. Joseph Island. And then there's also a set of American release sites just north of the Mackinac Bridge here in Michigan that were established in 2014. So what that tells us is that the parasitoids have either dispersed, from our St. Joe sites up into north in the Algoma region, or from the American release sites into this area. And so that's one of the questions we're going to get, we hope to get at with our field season in 2021, is looking at what's the spread in the Algoma region from these northern Michigan and Canadian release sites. So we want to set up some plots and look and see how far have these parasitoids spread in our area. And this builds on some work we've been doing in Southern Ontario that we started in 2018 and 2019, looking at spread from some of our oldest release sites. Next year, this year in biocontrol, what we hope to be able to do is to continue releases within Canada, including making up what we missed in 2020 at our sites in Ontario. Every year we have the goal to expand our release sites networks in Ontario and Quebec and the Maritimes. And so we've been looking at that, looking at sites, and we actually made it into the news in Pembroke recently. That's going to be one of the sites for releases in 2021. What we did do, because of the uncertainty about being able to go to the US to get the parasitoid allocation from our American colleagues, was we expanded the rearing capacity in Canada to supply release sites in the event that we still can't go to the States. So hopefully we'll have enough sort of made in Canada parasitoids to supply what we need for 2021. And then also we want to continue our spread and our impact assessments at our early release sites. And this is work we've been doing for the past couple of years. So just jump a little bit and talk a little bit about some advancements in ecology and detection and control. And this is just a photo showing here of one of our colleagues at the Atlantic Forestry Center, demonstrating a different style of emerald ash borer trap that John Sweeney and his team have been trying out in the Maritimes. And this is a green lingering funnel trap, and John has some data that shows it probably works a little bit better for collecting EAB, especially when it's and detecting EAB, especially when it's deployed high up in the canopy. Let's talk a little bit about some overwintering survival work we did. Folks who've been following the weather recently have noticed that might have noticed that we're perhaps due for a cold snap here in Eastern Canada. Another polar vortex event like we experienced in 2019, and one of the questions we asked following that polar vortex in 2019 is, what's the effect of that cold snap on EAB? Folks might remember from a couple of years ago, there was some work looking at how well EAB can survive Canadian climate, how well it can survive, what's its cold tolerance, and how that might affect the range of EAB. So we took those models, there's some work with Barry Cook and Remy Saint-Amand at our lab in Quebec City, and looked at predicted the survival of emerald ash borer using some of that overwintering tolerance data. And what we were able to show is that for that polar vortex in 2019 is that we probably had significant mortality through sort of northern Ontario into Quebec, and then in those populations in the Maritimes, but not in southern Ontario. And folks in southern Ontario might remember that 2019 as being cold. It wasn't very cold if you were an emerald ash borer. You probably survived. But this sort of got us thinking about what's this effect on cold, and what is it doing to these populations of emerald ash borer as it moves further north, and especially this population in Winnipeg, which obviously had been there for a while and has survived for a long time. And so what we did is we actually went and got some infested emerald ash borer bolts from Winnipeg with the help of colleagues with Province of Manitoba and the City of Winnipeg, and this work was taken on by Megan Dool, who's a postdoc in Brent Sinclair's lab at Western University, and they wanted to look at what's the cold tolerance of these western populations of emerald ash borer, because we knew it was surviving. And Brent had, with some of his grad students sort of early on in the emerald ash borer invasion, had looked at the cold tolerance of populations from around London, Ontario, and what they found when they looked at those populations around London, Ontario, is that those EAB prepupas, so the stage that most of the population spends the winter at, had a supercooling point of about -27 and experienced 90% mortality at -30 and 99% mortality at -35. And that's the data we use to do that map I just showed you earlier. So, EAB can take a fair degree of cold, but when you get sub 27, you probably should see some kind of mortality, significant mortality. So Megan repeated those experiments on EAB larvae and EAB prepupa with the Winnipeg population, and what she found is that Winnipeg EAB are extremely cold tolerant. So this is the sort of the survival at different temperatures when you expose to London populations, and these are Winnipeg populations. So they were surviving down to -45 and -50 in some cases for the prepupa. So that's extremely cold tolerant. And so some of the questions that we've been looking at following up on this work is, is this an example of the EAB evolving cold tolerance in response to selection by being, by living in Winnipeg? Or is it the result of, is this sort of some natural ability that the EAB has to survive extreme cold, and it just, it acclimates to, because of the Winnipeg sort of fall, winter? And this is some work that Megan is following up on. So just going to jump a little bit and talk about some other work that my colleague George Chaipoku has been doing. And this is a recent paper that George published with some folks at Great Lakes, looking at a new detection tool for identifying emerald ash borer on traps. So what George's tool does is it uses genetic tools to rapidly identify and confirm emerald ash borer on traps and in other samples. And the really cool thing about this is that it's a genetic tool, but it's something that you can do sort of in 30 minutes at about 65 degrees Celsius. So this is something that can be done, on a bench top in a fairly simple lab. The really neat thing I thought about this that really gets me interested is that it can detect the presence of emerald ash borer on traps where the insects have fallen off. And if folks are familiar with the big green panel traps that are used to survey for emerald ash borer, they sometimes will lose the insect depending on just by, because it gets warm, or if they're not checked early enough. And Jeff Fidgen has been working on this a little bit, looking at how often that happens. But George's tool can actually pick up evidence of emerald ash borer DNA actually being on these traps even after the insect has fallen off. And that's just what this picture is showing here. I think this stuff is really cool, and it's some interesting applications in other species like Hemlock woolly Adelgid, and looking for other invasive species on traps. One thing I want to talk a little bit about the FraxiProtect tool, and this is something that's been John Sweeney at the Atlantic Forestry Center has been working on with GDG and testing a product called FraxiProtect, which is a Bovaria bassiana formulation, and it's installed inside the bottom part of this of a green funnel trap like this. And the idea is that live emerald ash borer fly into the trap, fall through it, and instead of dying, they actually get contaminated by the fungus and then go off and carry it to other emerald ash borer. And this is work that Robert Lavallee at Laurentian started, and he's now retired, is picked up by John and the folks at GDG. And they've published a paper looking at this, the efficacy of this, and they've filed for registration in the States and in Canada. John had some experiments going in 2020, or planned some experiments in 2020 in Nova Scotia and New Brunswick, but those got interrupted because of COVID. And I think he plans to start that up in 2021. So this is perhaps another tool for managing emerald ash borer, especially probably in cities. So just change gears from talking about management of emerald ash borer and some of the ecology work we've been doing, and talk a little bit about some restoration work and what do we do after emerald ash borer has sort of gone through. And Donnie McPhee at the National Tree Seed Center in Fredericton at the Atlantic Forestry Center has been coordinating collections of ash genetic material, looking for basically establishing a seed bank, at the seed center for future replanting efforts or future genetic breeding efforts to try and put ash back on the landscape. And the map I'm showing is an area where it shows where the collections have been made with the past couple of years, and that's sort of the white circles. Those are the areas where Donnie's has already made collections. But the red areas are where he's still interested in receiving samples and knowing about sort of ash masting events, so they can go and collect seeds. And his high priority areas for the next couple of years is Sault Ste. Marie and around Thunder Bay for both black and white and green ash. So Donnie's contact information is there, and if folks are interested in participating in that, I encourage you to reach out to him. He's always looking for folks that are able to provide seed collections. So the final thing I'll talk about today is some of the stuff about deregulation of emerald ash borer in the United States. And some folks may have heard the news from about mid-January, where the USDA took the decision to deregulate emerald ash borer, what this basically means is that they will no longer regulate the movement of ash within the United States because they're for various reasons, they've decided to take the resources they were putting into that part of their EAB control program and redirect them into things like biological control. And so since they took that decision, some of the states have said that they're going to still have their own controls or start their own controls on the movement of ash and ash firewood and this kind of thing. But this decision and that the USDA was interested in doing this has been sort of on the books for a couple of years. I remember hearing it about it almost 2 years ago now. And so in light of this probably happening, colleagues in Dan McKenney's group at Great Lakes were asked by CFIA to see if deregulation was also appropriate for Canada. And so they did a bunch of work and they published this report, and Emily Hope is the lead author. And what they found is that even with, basically asking the question, was deregulation of emerald ash were also appropriate for Canada? And what they found is that even with a minimal effectiveness in their regulatory efforts slowing the spread, the regulation in Canada is likely efficient. But it was still likely that EAB will continue to spread much further across the country regardless of that regulation. And there's a lot in this report. I'd encourage you to go check it out. And I'm told the authors are planning to do an online webinar with the Invasive Species Center in the next few months to sort of talk about, going more in depth about the study and what they found. And I'd encourage you to keep your eyes open for that. So just a summary. So basically, just summarize the talk real quick. So the regulated area for Canada slightly expanded in 2020. Our biological control program is slowed by COVID, like just about everything else, but will continue in 2021. We have a lot of potential for ongoing work to look at impact and risk prediction, detection, and mitigation for EAB, and some of the things we've learned about EAB are helping us get ready and manage other species, especially like Hemlock Adelgid, which is something we're thinking more and more about. And then the collection of these genetic resources for breeding and replanting is ongoing, and there's a lot of interest in that work. So,
Michigan. I'll be going over hemlock in Michigan, HWA, its known distribution, and our statewide strategy. There are over 176 million hemlock trees growing in Michigan, with 1,000 more being planted in landscapes. Much of our hemlock resource consists of relatively older trees. Seedlings and saplings tend to succumb to deer browsing, and hemlock seeds are easily damaged by drying. In one study, 60% of the seeds were severely damaged after only 2 hours of drying, and 80% died or did not recover after 6 hours. Hemlock is shade tolerant and can live in a forested understory for 25 years or more. It provides critical winter cover, food, and habitat for several bird and mammal species like deer yarding areas. Loss of hemlock will have a huge negative impact across the landscape, especially in riparian zones and our critical dune habitat. Loss of shading along our rivers and streams will lead to increased water temperatures, which will negatively impact our cold water loving fish species like trout. This is our hemlock range in Michigan. The darker the purple, the higher the density of hemlocks. HWA is an insect that, as an adult, pierces its host and sucks out nutrients. It was introduced from Japan, first identified in the 1920s in Western states, and it's interesting to note that Western species of hemlock is resistant to HWA. It was found in our eastern states in the 1950s, and an infested tree will usually die within 4 to 10 years. HWA has an interesting life cycle. It has 2 generations per year, and those stages can overlap. They are reproduced parthenogenically, so they are all females. The insects and eggs are protected by the ovisac, which is a white waxy material secreted from glands on the Adelgid's body. The Pregridian stage feed from late spring to early summer. In Michigan, each female lays up to 50 eggs, and those eggs hatch into cystins. The cystins feed from late fall to early spring, and in Michigan, females lay up to 200 eggs. Those eggs hatch into Pregridians. You'll note that because the cystins feed from late fall to early spring, there is a long time that they're able to build up the ovisac, so that white waxy cottony material ends up showing up really well during November through March, which is our best times to survey. This slide shows different stages of HWA. There are the eggs with ovisacs, crawlers, and on the bottom left, you can see the up-close picture of the mature Adelgid with its stylet bundle kind of rolled up, but that single line in there is what it inserts into the plant to feed from. The bottom middle picture will show at the upper top arrow pointing to the Adelgid itself. And you can see going down through that tiny little white line being pointed to by the two bottom arrows, that's the stylet bundle in the tree itself. On the bottom right, you'll see that even that very first instar will end up producing that white woolly ovisac. On the tree itself, we look for the white woolly ovisacs at the base of the needle on the twigs. And this placement is important because it'll help us identify HWA from other confusing lookalikes. So again, at the base of the needles on the twig. Crawlers are the only mobile life stage. They are carried through wind on birds and other animals and through the improper disposal of infested branches. This shows the time when crawlers are most likely to be spread. Care is taken to reduce the spread during survey and treatment by having a set of field clothes which can be inspected and removed at the end of the day. The clothes can then be washed and dried. I usually get questions as to whether birds can really move crawlers or not. This is a paper I came across showing that while the study design cannot confirm any long-distance dispersal events occurred, the results show that the transfer of HWA crawlers from branches to birds occurs most often during periods of peak crawler abundance, and that crawlers are capable of actively moving off birds and settling onto uninfested hemlock foliage. Another question I get is whether cold temperatures will kill them. Research in the northeastern states show that there can be up to 90% mortality if temperatures drop below negative 22 degrees Fahrenheit or negative 30 degrees Celsius. This is helpful to areas in northern Michigan and the Upper Peninsula, but snow cover insulates the branches, and with 2 generations per year of all females, it means that populations can increase quickly. Every little bit helps though. Here are some lookalikes that can be confusing when looking for HWA. You'll notice the spider egg sacs on the bottom. Those are very difficult at times. So remembering that HWA is located at the base of the needle on the twig is very helpful. Another interesting insect is elongate hemlock scale. It's an invasive that has been observed on trees in areas infested with HWA at a relatively high incidence. So if you come across a tree that has a lot of this, you might want to take a little bit extra time to survey, see if there is any HWA on the same tree. The health of the tree can be an important indicator of HWA presence. Because it feeds off of carbohydrates and nutrients, it will stress the tree, slowly killing it over time in as little as 4 to 10 years, depending on the size of the tree and the density of the Adelgid. So it's important to keep an eye on unhealthy trees and their proximity to previous introductions, active infestations, and nurseries that handle hemlock. On the left, you'll see a healthy hemlock tree with green, dense foliage versus an unhealthy tree on the right, showing the grayish yellow needles and loss of foliage. Again, beautiful, lush green foliage with new growth on the left, and on the right, you can see the grayish yellow and even browning of needles and their low density foliage. This map shows the distribution of HWA as of 2019. We just added a new county, Mason, in 2020, making it the northernmost known infestation in Michigan at this time. HWA was first detected in Michigan in 2006. Because this area has a high density of hemlocks, multiple treatments, and numerous surveys have been conducted since it was found. HWA has not been detected here since 2010. Note that Mason County is now the northernmost location with known HWA. So we're going up here, Mason County, and then the infestations actively occurring right now along that lake shore. The current known extent of HWA in Michigan runs approximately 110 miles along the shoreline of eastern Lake Michigan. Again, the northernmost point is in Mason County, with more positive locations being found this survey season in and around Ludington State Park. The Pentwater State Game Area is the first positive found on state forest land. So up here is our northernmost extent. This is at Ludington State Park, and then we've had them located all along the shoreline again, with some extending a little further east down here in Ottawa County. To address the ongoing concerns, a statewide strategy was created in 2017 and contains 6 subgroups. Data and technology includes Forest Resources Division's Resource Assessment Unit, having created our detection surveys app using Survey 123. It begins by collecting information on the overall location of the survey area and then focuses on each point. One point reflects 1 acre surveyed. Up to 30 trees per acre are surveyed if available. This number allows for good coverage of the area without bogging the survey down by trying to look at every single tree. You'll note that one cool feature is down here we can take photos of any questionable sighting that we find, and then that can be loaded and looked at right away once downloaded. We're lucky to have a large amount of collaborative partners helping with surveys. These entities are vital to early detection efforts, and the more entities and folks on the ground that are out there looking, the quicker we can find any new sites. The state of Michigan has 2 departments actively working on HWA. In 2018, the Forest Resources Division received funding to create a forest health response team. Another forester and myself focus our efforts on state forest land, which is separate from state park land. We also look at federal lands and assist others as needed. Park and Recreation Division focuses on state park land, and the Department of Agriculture and Rural Development reviews suspect samples and maintain the internal and external quarantines. Detection surveys are quick and broad. They occur in proximity to known infestations, and they are focused along the Lake Michigan shoreline due to the lake's moderating temperature effect, which increases the likelihood of HWA survival, and because migratory bird patterns tend to follow the lakeshore. Delimitation surveys are intense and specific. They occur in areas with known HWA to determine the extent of the infestation. These surveys begin at the HWA positive's location and are conducted until HWA is no longer found. Hemlocks are selected on the basis of having branches within reach, but visual inspection of higher branches is important too. And note that a single tree that is positive for HWA establishes its presence in an area, and delimitation surveys begin. We'll also carry pole pruners with us to help reach some of those higher branches where we can. I wanted to include this slide from our West Michigan SISMA because it shows a crew member during a delimitation survey. Note that all trees to be treated are tagged and the number recorded to correspond with the DBH of the tree. And I just love this picture because the tape is straight across on that tree. It's not raised up or down on one side. And so as a forester, this is just a beautiful, beautiful picture of a tree being measured. So you can see West Michigan SISMO worked on over 800 properties, completed nearly 3,000 acres of delimitation surveys, and inspected over 60,000 hemlocks for HWA. Survey efforts occur with the goal of mapping the majority of HWA in Michigan effectively given limited funding, with the knowledge that we will not be able to capture all infestations and to develop a long-term management plan to slow the spread and reduce impacts. This shows the location of our 2019 and 2020 detection surveys, showing 9,403 acres were covered through our cooperative efforts. The red points show 2020 and the blue points show the 2019 surveys. Treatments are prioritized based on the northernmost extent and work southward back towards the positive. Cooperative Invasive Species Management Areas, or CISMAs, coordinate treatments and surveys on private land, but they must get landowner permission beforehand. DNR Parks and Rec Division and Forest Resources Division coordinate on state lands. All hemlocks within 800 feet, or approximately 244 meters, are treated with the goal of local eradication and containment. 2 active ingredients are used for treatment. Dinotefuran is applied as a basal trunk spray and persists for 1 to 2 years. Imidacloprid is applied as a tree injection or basal spray and persists for 3 to 5 years. Cost and per acre requirements limit chemical use. Both can be applied as a soil drench, but due to the proximity to Lake Michigan and our critical dunes habitat, we are avoiding it over leaching concerns. Both can be used together to provide both immediate and long-term control. Crews are out every day treating or surveying for HWA. They work on steep slopes and have to hike in most spots to access their sites. These are very difficult working conditions, but they are troopers. The totals for Parks and Recreation Division show a total of 30,558 trees treated since 2017, but the really crazy number to me is the amount of inches treated. 187,738 over 4 years. These are the numbers for the West Michigan SISMA. They've treated over 2,500 acres on 586 different properties. And remember, they have to do the pre-work because they're dealing with private lands, so they have to make contact and get approval before being able to step on any land that they survey or treat. Their total trees treated were 46,563. So here's the treatment summary for the 2020 season and overall since 2017. Note that HWA has not been detected on any of our federal lands in Michigan, and the asterisk by the state shows that HWA was detected at our Pentwater State Game Area, but was in the fall, so treatment wasn't being done. It will start this treatment season coming up. Overall, since 2017, a total of 520,623 inches have been treated, which is 13,223.8 meters. So again, a huge collaborative effort by everyone, all of the partners. So thankful for so many entities that we have working together. Outreach and education are essential to bringing awareness of issues to the public. Through our media efforts alone, we have been able to reach approximately 600,000 people since 2018. In pre-COVID, in-person events and trainings have reached 4,654 individuals. The DNR's Marketing and Outreach Division has been a huge help in reaching these numbers. Our subgroup also created this two-sided little ID card for hemlock tree identification, as well as hemlock woolly Adelgid identification and reporting options. So this is just a little pocket size individual card that we've gotten huge positive responses from. Continued research is occurring on temperature mortality rates, chemicals and their application rates and methods for efficacy, trapping methods, a qPCR testing protocol for finding HWA eDNA from traps to detect if HWA is in the area, phenology, survey and management, and hemlock distribution models. The HWA exterior quarantine was implemented in 2001 to mitigate the introduction of HWA into the state from other infestations in the country. You'll notice our latest was revised as of September 10th, 2020, as more information is being gathered. The interior quarantine was implemented in 2017 to mitigate further spread within the state. This is our definitive regulatory tool. It regulates the movement of hemlock out of, into, and within the infested counties. This shows our interior quarantines list of high and low risk articles. High risk is hemlock nursery stock, hemlock yard waste, tree trimmings, and branches and limbs. And this is a photo of infested branches in one of our HWA active population areas. So easily spread by landowners that don't know what it is or don't know what to look for. A few different low risk articles: lumber, logs and firewood, composted, chipped, shredded, grounded parts, articles transiting during low risk period, again when those crawlers aren't present. And that's the end of my presentation. Thank you all very much.
Excellent. I hope that you are all able to see that. So we do have Cheryl Nelson on the line. Oh, I'm sorry. Now I'm doing another. My apologies. I had another YouTube video start playing in the background after Cheryl's ended. So we do have Cheryl on the line, as well as Dr. MacQuarrie from his presentation previous, and we're able to take any questions.
I'll start with some questions directed at Chris MacQuarrie. So what is the distance between the two release sites and the findings in the ash bolts last November up north?
Yeah, it's so from the closest site, sort of south of Echo Bay, it's 35 kilometers to, as the crow flies, to the release site on St. Joe's, and it's 70 kilometers to the American site. And to Sault Ste. Marie, it's about 60 kilometers, and from St. Joe's, and about 70 kilometers out to the American release site.
Thank you. And are they still looking for seed from black ash in southern Ontario?
I think I don't know is the short answer. Donnie would be the best person to ask, and I think Nicole put his contact information in the chat. Okay, cool.
Yeah, it is.
I had a question for Cheryl, actually, after Cheryl's presentation. Cheryl, I'm just wondering if you know of any kind of collaborative efforts that are occurring for the HWA very close to the border. That's obviously troubling to Canadian agencies as well. I'm wondering if there has been some collaboration between countries and different agencies on managing and detecting that.
There's nothing that I'm aware of. I'm sure individuals are talking. So it is something where you can get a hold of me. I can get more information on that. But right now, since it's not in Michigan in that area. We're focusing our efforts on the southwest side of the state.
Excellent, thank you. Taylor, I see your hand up. Potentially you want to weigh in on that?
Yes, thank you. So I'll just add to that there is a Michigan interagency forum on forest invasive species. It's been around for a number of years, and I sit on that as the Canadian representative. And there's lots of contact as well with people like Chris and others with our colleagues in Michigan on a regular basis. So there is international collaboration, cooperation, and when it comes to things like the quarantine, regulatory controls, and research, we are involved in that quite a bit.
Excellent, thank you.
So just in case people aren't up to date with what is in the chat here, there was a question asking if biological control is available to private landowners, and Chris did write a response saying, we're always looking for release sites and encourage anyone who may have a candidate site for release to get in touch. However, as far as he's aware, they are not available for private use. So are there any further questions? If not, I think there is 2 questions here for Allison. I do see your response here, but do you want to just quickly reiterate the answer to these 2 questions? Does one need a pesticide license to apply this in Ontario? And what is the efficacy compared to glyphosate applied in the same method?
Sure. So yes, you do need a pesticide applicator's license. So you need a landscape or forestry type exterminator license, and that's required to purchase the product and apply it. And just for general information, Lallemand chondro is a Class C pesticide. And then for the next question about glyphosate, our trials were looking specifically just at the overall product efficacy of Lallemand chondro. Not in a comparison trial against other control methods, except for, of course, a control to compare our product efficacy against.
Great, thank you. I do see a question that has come in here for Cheryl. Are there differences? Oh, hold on. It just disappeared on me. So
that was my bad. Sorry, I'm getting used to Zoom here. I can read it out because I moved it to the answered section. It was from Ronwu Winter wondering if there are differences in efficacy between the basal bark dinotefuran and imidacloprid injection treatments for HWA.
Yeah. I guess I'm not sure of that. Dave, if you are, please jump in, or Simeon, because that's something where we use both individually and together. So I know that they're both very high.
Chris, I see your hand up.
Yeah, the dinotefuran, so in the States, they can use a tank mix, an application of dinotefuran and imidacloprid together, sprayed on trees, and you get the dinotefuran sort of this very quick knockdown of HWA, and then you get sort of longer control with the imidacloprid product, and that's what is used in the injected product. In Canada, we don't have access to dinotefuran.
Thank you. And another note, we inject trees that are 5 inches and larger, and then we'll do basal bark sprays on anything under that. Seedlings, saplings, full size under.
Yeah, and in Canada right now, the only product that's available is, so I guess there's 2. There's the injected Imajet product, and Bioforest's triazine product, I think, is now registered for HWA, and we've looked at, we've started some work looking with John Sweeney at Atlantic, and we've started some work in Ontario looking at the sprayed product, just the imidacloprid, and I found out recently that they're looking at an emergency registration for the sprayed imidacloprid for Nova Scotia. So that's what we have for access in Canada.
Great, thanks for clarifying that for us. I'll wait another minute here just in case anybody else has any follow-up questions. And if not, we will end the session. But I just wanted to quickly thank everybody who decided to join us here today. I know this was long awaited for many people, as we had planned this for last April and had to postpone as the pandemic occurred. So I really appreciate all of the speakers coming back to do these presentations this year, and hopefully next year we can meet again in person and have this as a full day event or longer. As we go on here, if you don't mind and you have comments or feedback on how today went, and if you've attended Forest Health Reviews in the past, please feel free to email us and reach out and let us know what you're thinking, what you'd like to see in the future, what you liked about this. I think that we will continue to offer, as we enter this new phase in our world, the virtual option to participate in these in the future as well. I just want to thank Nicole and Forest Ontario too for helping us get this going again. I know it was very last minute, but the fact that we had almost 150 people or more with us today, that's really great. A week and a half to register. We really appreciate everybody participating. And I just, as a couple questions are coming in here, there is a question of when the 2020 Forest Health Conditions Report will be available online. I don't remember what Dan said there. I feel like it should be soon, but he had to go on to another session. So please feel free to contact him directly if you have any questions.